Welcome back. I know we look sparse, but I think people start filling in. And it would be a shame if this whole place wasn't filled, because Christine Houghton is a leader in this field of nutrigenomics. I had the pleasure to hear her speak a couple of years ago at our first half a day or day of precision medicine, and it was just an amazing lecture. I learned an incredible amount, and I'm looking forward to this one as well. The notion that nutrition can influence oxygenation of genes, changes in genes, is something I mentioned in passing this morning, and you'll learn a lot more about. Christine is currently engaged in research at the University of Queensland in Australia, where she's investigating bioactive nutrigenomic phytochemicals with significant clinical potential. She is the author of Switched On, Harnessing the Power of Nutrigenomics to Optimize Health, and she's here to talk about how clinicians can incorporate concepts into their practice. Welcome.
Yeah, thank you. Just give me half a tick. So it's a pleasure to be here. This is the third year I've been at an AMMG conference in Las Vegas, so it's a very great pleasure to be back and meet new people and reconnect with familiar faces. So I'm going to suggest to you today that the dual sciences of nutrigenetics and nutrigenomics have the potential to reshape the nutrition landscape as we know it. The one-size-fits-all diet solution is by necessity what public health authorities recommend. And whether you like this choosemyplate.gov approach or not, I think you'd have to agree that if all of the people in your country and mine were to even eat according to this recommendation, we would have much less disease than we see now. But as you know, one size does not fit all, so we have to keep trying, and eventually we'll find the perfect fit. And that's where the era of personalised nutrition fits in. So we have been looking now for the last decade or just a little more at this concept of personalised nutrition as it relates to an individual's nutrigenetic profile. But we're very early in our stages of development with this, and there are significant challenges in translating what we know of SNP data into personalised advice. And there are many SNP diet studies that don't show a cause-effect association. And we'll talk more later about why that might be. And it's also important to know that classical genetics is associating the genotype with a disease risk, whereas in personalised nutrition, rather than personalised medicine, the personal data relates to the optimal diet for a given genotype, and with the goal of reducing disease risk. And I think consumers in general would welcome such information. So what is nutrigenomics? What is nutrigenetics? Well, as I see it, it is an interweaving of a number of different disciplines. And if we start on the left with nutrition, classical nutrition as we know it, which is underpinned by biochemistry, and then we look over on the right at nutrigenomics, nutrigenetics, genomics, all of those factors combined contribute to human health. But nutrigenomics is not simply a combination of nutrition and genomics. It really is a new discipline in its own right, and while it embodies all of those principles, it is evolving in its own way. Nutrigenomics arguably is driving the paradigm shift in the practice of nutritional medicine, and it's certainly adding an additional dimension to the concept of personalised medicine. Well, who is the nutrigenomics community? We now have a couple of international bodies. Up on the top left you see NUGO, a European organisation, and ISNN, the International Society of Nutrigenetics and Nutrigenomics. And populating that screen there now are a number of journals which are reporting the science in this area. So we talk about omics, and I think sometimes the complexity of omics can be quite confusing, but essentially we start with a diet, we end up with a phenotype, and in between all that there are a number of omics which we'll investigate in just a moment. We talk about metabolomics now. I mean, what does that mean? What is a metabolome? So if we look at this diagram, we see that genomics is really just a reflection of what is within the DNA. We transcribe that to the messenger RNA, that's transcriptomics. We produce a protein, that's proteomics, and we end up with some metabolites, and that's metabolomics. So in a nutshell it is, the genome is what can happen, the transcriptome is what appears to be happening, the proteome is what makes it happen, and the metabolome is then what has happened and what is happening. And of course now we've got the microbiome, and you know, the microbiome has taken the scientific community by storm. And those of us who've been practicing nutritional medicine for a long time probably not surprised at the importance of the microbiome, although we gave it a different name in days gone by. But we really have to extend our definition now, and here's a paper from 2011 that says the extended nutrigenomics understand the interplay between the genomes of food, gut microbes and human host. So we now have another layer of complexity to deal with. So there are many influences on that phenotype, and we're looking at genetics. So what did we come to this earth with? Epigenetics, so we have some epigenetic changes, largely based on methylation changes, but also other chemical events like acetylation and others, which modify the genome but are not hard-coded into the DNA. And of course the environment has an enormous effect on what's happening. So if we're looking at the nature versus nurture story, we have all of those elements shown here. All of that combined generates the phenotype, which becomes you and becomes me. So if we're looking at doing nutrigenetic testing on our patients, we are largely looking at SNPs and mutations and so on. So that's the realm in which we work. If we're looking at a primary intervention focus, we're largely looking at nutrition, perhaps pathogens, drugs, toxins. And in the realm of epigenetics, we have a limited ability to clinically assess or intervene here. But it's coming and it's changing. As an earlier speaker was mentioning, this concept of SNPs is really coming about because we've essentially got spelling mistakes going on in the DNA, and some of those spelling mistakes, as he was pointing out, are fairly irrelevant. They don't change what's happening. Some of them are very significant. So in this case here, we've got an AT nucleotide pair which has been converted to something else, and in the process that is sufficiently significant that we've altered the protein which is finally formed. So in altering the chain of amino acids that have made up that protein, we often also change the conformation of that protein, because interactions between amino acids can change the shape of the protein. As a consequence of that, if that protein happens to be an enzyme, we can significantly alter the enzyme's activity. And hence, if we're talking about a SNP, which has a certain expected level of activity as an enzyme, and then we see a downregulation of that enzyme activity, we now are going to have altered function in all of the pathways that are governed by that enzyme. This is probably reviewing some of the things you already know. So not all single nucleotide changes are SNPs. And to be a SNP, we have to have 2 or more versions of that sequence present in at least 1% of the population or the general population to be classified as a heritable SNP. SNPs occur throughout the human genome, about 1 in every 300 nucleotide base pairs, which translates to an enormous 10 billion SNPs within the 3 billion nucleotide human genome. So it's no wonder that there are so many variations between us. So, quite simply, as I'm sure you know, a genotype is just the sum total of our information in our genes, and the phenotype is the expression of that information. So how does this work in practice? If we had a nutrigenetic profile that told us that a particular patient's tumour necrosis factor, TNF-alpha gene, had actually produced a variant of that gene, so a SNP, and this particular SNP, for example, has a twofold increase in TNF-alpha transcription, we're now going to increase the transcription of TNF-alpha, so we're going to get more inflammation within that patient. So those are the sorts of things which we can look at clinically if we have that sort of information. So this is just one of the pro-inflammatory genes, but the same applies to all of the other cytokines that you might have information on, or genes coding for cytokines. And essentially the gene is acting on the cellular environment and now we're getting increased inflammation. Conversely, nutrigenomically, where we can intervene, we know that omega-3 fatty acids at a certain dose can down-regulate TNF-alpha expression. And so now we have a nutrient or a bioactive compound which is acting on the gene. So essentially we use nutrigenetics to test the patient and find out where the patient's variant genes are, or SNPs, and we use our knowledge of nutrigenomics to know how to intervene to change the expression of that gene. So in this case, with a patient whose TNF-alpha has been upregulated, we can use omega-3 fatty acids at a certain dose to downregulate that closer to what the normal variant of that gene would reflect. So I talk about translational nutrigenomics. We teach translational nutrigenomics as something which is clinically relevant. So I went to a conference, a NUGO conference in Italy last year, and it was a week long, and it was the annual Nutrigenomics conference. But I was a little disappointed in that. Exciting as the science was that was presented, it didn't have a lot of clinical relevance, and it was out of that lack of clinical relevance that I felt there was a very great need for clinicians to get a better understanding of what we actually do with this from a clinical point of view. So the way I see it is this: we start with some knowledge of the nutrigenetic profile and the nutrigenomic interventions we can use. We have to tie this into the nutritional biochemistry, because every gene codes for a protein, probably an enzyme, and every enzyme is involved in a pathway somewhere. So if I understand what that enzyme does normally in that pathway, then I can begin to understand that if that enzyme is functioning below normal activity, then I can expect that there will be some interference in that pathway. And I can then, if I understand the biochemistry, begin to understand how that is impacting on the health of that person sitting in front of me. We talked too about test pairing to establish function. So you start with your nutrigenetic profile and you intervene in some way nutritionally, but then how do I monitor what's changing? So test pairing is really using a lot of the tests you already use to consider what's happening from a nutritional standpoint or a biochemical standpoint. But if you don't do that, of course, you've got no way of monitoring the gene expression that you're expecting to change. Sometimes in limited circumstances we can intervene epigenetically. We look closely to it culinary genetics or culinary genomics. Let's not forget that nutrigenomics is about food, and man and woman have existed on this planet for who knows how long on the strength of the food environment that we've had access to. So long before we'd had access to supplementation or any of the knowledge involving this, man has used food to change gene expression. And then, of course, we may need to use nutritional medicine, go to a higher level, use a higher dose of whatever the intervention might be. So that, in a nutshell, is what I call translational nutrigenomics, from lab bench to clinic. What nutrigenomics is not, I think, is very important to say. There's a popular trend right now to think that the phenomenon of methylation is nutrigenomics. It's not. Methylation is one cog in a very large wheel that makes up human biochemistry. And unfortunately, there's this trend now that looks at the MTHFR polymorphisms and the SNPs associated with methylation pathways as if that is all one needs to know to deal with nutrigenomics. And what disturbs me greatly is that there are now a large number of consumers who are having these SNPs assessed and find that they have an MTHFR defect, for example, and as a consequence of that, they then assume that everything that's ever been wrong with them is happening as a consequence of that gene. So if I've been onto some of the forums where consumers are discussing this, and I'm just aghast at some of the anguish that people are going through. Here's one. This is just verbatim straight off someone's forum, from an MTHFR forum commentary. She says, maybe someone can offer some help. I've recently been diagnosed with homozygous 667T, which is the MTHFR, by my naturopathic doctor. I was taking B12 complex plus with 400 micrograms of B12 and 400 micrograms of 5-methylfolate and other Bs. Was on it for 2 months and felt tired, heavy feeling and jittery. I went off all the supplements for a week, was told I should try a fifth of a tablet for 1 week, go on to 1,000 micrograms, etc. Then she says, it kind of feels you're hanging. What to do next? I was then advised, if you don't feel well taking half a capsule, stop taking it. You're either receiving sufficient methylfolate at this time or you're not yet prepared to take it due to other mutations present or other health issues worsened by methylfolate. She says, okay, so what is it? I'm receiving enough, I have other mutations, or what? Very confusing. Oh, it's been 3 days on the 1,000 micrograms B12 and already getting that tired, jittery feeling again. This is not once, this is often I see this. It is very concerning. And from my perspective, the problem is lying in the fact that we need to put methylation in its proper perspective. I look at cell defence as really the core of what we're trying to influence. If we can influence every cell in the body to function at its optimal level by looking at all of the various aspects that govern cell defence, we are not likely to single out one particular focus like methylation and cause problems. So the way I see it, I put cell defence in the middle. We're then looking at important functions or processes which are necessary to govern cell defence. There's redox status, the inflammation and immune function, energy production, detoxification. And I put methylation out on the side because it actually has a dual role. It works in standard biochemical pathways, but it also has epigenetic effects. So that's why it's sitting out there on its own. Back to the one size fits all. Let's just have a look at an example of the dietary guidelines. If we take a bioactive food element and we're looking at the phenotype, which is the end point of all this, let's remember that in the middle of all of this are our various omics and processes. I want to try and simplify that and make it clinically relevant. So I'm going to use an example. Practically everybody drinks coffee or has had coffee at some stage. If coffee disagrees with you, you probably know it because you probably can't get to sleep if you have it late at night. So you probably suspect if you have variants. But this paper here from 2005 probably creates more confusion than it solves problems. So the abstract summarises the opinions and it says boiled coffee is associated with increased cardiovascular risk and increased plasma cholesterol. But moderate daily filtered coffee is not associated with risk. Significant antioxidant content, which is an inverse effect on type 2 diabetes risk, and coffee consumption is associated with increased homocysteine. Is coffee lowering B6? Well, I don't know what you make of all that because I can't make anything of it. But let's try and sort through it. So how much caffeine am I consuming daily? Well, the FDA in its infinite wisdom says that 400 milligrams a day is safe for most adults. And so if you're having 4 cups of brewed coffee a day, that's your 400 milligram limit. Of course, unless you go to the local Starbucks and get whatever these large Americanos are that haven't reached Australia yet, I'm glad to say, you're getting 300 milligrams in that 1 cup. So if we're trying to work out what's going on here in terms of who reacts to caffeine and who doesn't, we need to look at the CYP1A2 gene, because it's responsible for about 95% of caffeine metabolism, and it's widely varied between individuals. So if we start with the biochemistry of coffee and its potential impact on health, we see that at that 400 milligram a day dose, caffeine breaks down to 3 metabolites: paraxanthine, theobromine, theophylline, doesn't matter what they are. But CYP1A2 is the rate-limiting enzyme in that pathway, so it's pretty important that we understand what it's doing. And interestingly too, its activity is upregulated by smoking. So what does this mean? If I have the CYP1A2AA genotype, I'm the fast metaboliser. But I might equally have SNPs, which are the AC SNP or the CC SNP, the heterozygous and the homozygous, and in both cases I'm going to be a slow metaboliser. So if I have a look at this inducibility graph here, I see that depending on my genotype, I'm going to be a fast metaboliser or a slow metaboliser, and there's around about a 4 to 5 fold difference depending on which SNP you have. 50% of the population are slow metabolisers, so if you look to the person to your side, one of you is probably a slow metaboliser. Now, does it matter whether I can't sleep at night or not? Probably not. I just don't drink coffee during the later part of the day and it's not going to bother me. However, we now know that coffee affects myocardial risk, and maybe that puts a different slant on it, and maybe this is pretty useful information for you and your patients to know. So when you look at this on a population basis and you look at the 4 cups a day that the FDA says is safe for us to take, you can see there that the more coffee you're drinking, the greater the myocardial risk. But the problem with this is, this graph here is just across the entire population. Now let's look at something interesting. Now let's pull apart the SNPs, the AC and the CC SNPs here, and now we see we've actually significantly increased the risk of myocardial infarction, about 4-fold, in people who are the slow metabolisers. So maybe it's heading off to the decaf again. So this is just an example of something that I think is pretty simple exercise. Everybody can understand it and it's pretty useful information. But there are those who would say that we're not really ready yet for nutrigenomics. You're going to have to make your own mind up at the end of this talk. So there are lots of papers now talking about the role of nutrigenetics and nutraceuticals. Is it the next wave riding on personalised medicine? This one says, do we know enough? A scientific and ethical analysis of the basis for genetic-based personalised nutrition, that's 2013. The following year, prescribing personalised nutrition for cardiovascular health. Are we ready? Well, are we ready? What do we need to consider to decide that? So the first thing we have to understand is which SNPs have actually been scientifically validated. So there's an important consideration we'll come back to. Which SNPs, validated or not, have clinical utility? For example, the taste genes have been scientifically validated, but they tell me whether I detect bitter tastes or don't detect bitter tastes. Interesting, clinically useful, may not be. Some labs, unfortunately, are making exaggerated and incorrect claims about what these SNPs mean, so that's something to be aware of. And the genetic variation is shown to affect food components, so depending on our nutrigenetic status, those SNPs can determine how we take up food, whether our transport systems are working properly, whether we can metabolise and eliminate metabolites effectively. All of that has a bearing on cell function. There's also implications now for the modification of recommended dietary intakes. And clinical genetics, we need to differentiate, is using genetic information to calculate disease risk. We are talking about nutrigenetics here. We are not talking about the monogenic diseases where we've got one disease, one SNP, one disease. We're really talking about nutrigenetics, which is operating at a level of influence over a whole host of cellular processes. Here again is a differentiation. If we look at atherosclerosis, on the left we see classical genetics tells us about familial hypercholesterolemia and tells us about the LDL receptor gene, ApoB, and a few others. Whereas, in terms of nutrigenetics, we're not so interested in genes that we can't modify, we're interested in ones we can modify. And genes in terms of those which can be modified by diet, lifestyle, physical activity and so on. And those modifiable effects can change redox balance, inflammatory processes, detoxification processes, methylation and others. So there is a difference in what we're doing with classical genetics and what we're doing with nutrigenetics. Here's a chart from 2014. A researcher has listed some of the validated genes. I'm just going to talk about a couple of these today. MTHFR, of course, is one of those validated. SOD2, superoxide dismutase, the manganese-dependent enzyme, which is prolific in the mitochondria because the superoxide radical species is generated in large quantities in the mitochondria, and so SOD2 is the antioxidant enzyme that is designed to regulate that at its source. CYP1A2, we talked about that with the caffeine. And we're going to talk about a couple of phase 2 genes in the glutathione S-transferase family, the GST family. So if you were to look at a particular nutrigenetic profile, this is just an example of one. So we're looking at the MN-SOD gene here, and if we just look at this in the patient format report, we see that this person had the beneficial or the normal variant of MN-SOD. But MN-SOD takes superoxide and converts it to hydrogen peroxide, which then goes to water and oxygen and gets rid of it. The hydrogen peroxide, which is the product of that MN-SOD, is a toxic reactive oxygen species in its own right. So in this person's case, this person is not successfully able to metabolise the hydrogen peroxide down to its water and oxygen as successfully. So what this means is there's likely to be a tendency to accumulate H2O2, which is a highly mobile reactive oxygen species which travels readily through membranes and travels around the cell and wreaks havoc elsewhere. So while this person might feel happy that he or she has the normal variant of the MN-SOD, they have the heterozygous variants of the GPX1 and the catalase. So there are problems with this genotype or phenotype. What else do we need to know about MN-SOD genotypes and risk? So this is just looking at a single gene now, not combinations of genes. So in this case, MN-SOD genotypes containing a certain variant were associated with a 1.5-fold increased risk of breast cancer compared with those with the homozygous wild type. And this finding supports the proposal that MN-SOD genotypes may modify individual breast cancer risk. We'll come to some other examples in a moment where we're looking at combinations of genes which give us a much better understanding of what's happening. So this is a 2015 study of another paper now that says MN-SOD polymorphisms strongly associated with the clinical significance of prostate cancer in men older than 69 but not in men younger than 70, suggesting that oxidative stress may be involved in the progression of the disease. MN-SOD may be a clinically useful marker to predict the potential of progression of prostate cancer. So if you know your patient's SNPs for these primary upstream genes which are acting right at the origin of the oxidative stress within the mitochondria, you have a much better opportunity to gauge their likely risk of disease expression. So here's another one. This paper that is entitled Diet, Genetic Polymorphisms, Detoxification and Health Risks from 2007 is talking about diet being a mixture of carcinogens, mutagens, and protective agents. And so every mouthful of food we eat is probably a combination of all three. All are metabolised by the detoxification enzymes. So it's important to realise that not only are known toxic compounds metabolised by these phase 2 enzymes, but some of our food elements are equally. And I point this one out in relation to broccoli, because if you have polymorphisms in the GSTM1 and GSTT1, which are glutathione S transferase by name, if you have the null genotype, you produce none of those enzymes. And you might think that's a disadvantage, but those deletions, in fact, confer enhanced protection on people eating cruciferous vegetables. Why? Because sulforaphane, the bioactive compound that's derived from broccoli, gets metabolised by GST, and if the GST is limited or absent, then the sulforaphane compound stays around in the blood and in the cells for much longer. And in particularly in bladder cancer patients has an enormous benefit, because those sulforaphane molecules, which are retained for several hours within the bladder, confer protection right there locally in the bladder. So not all SNPs are a disadvantage. The other thing that's important to remember when we're trying to enhance gene expression is most enzymes come with a cofactor or two. And if we're talking about SOD, the SOD form that's in the cytoplasm is actually copper and zinc dependent, so that enzyme is inactive without adequate copper or zinc. The mitochondrial form we've already mentioned is dependent on manganese, and the extracellular form again is dependent on copper and zinc. So those trace elements are absolutely essential for enzyme function, and I think we sometimes forget that. In terms of getting rid of the hydrogen peroxide, the GPX and the catalase require both selenium and iron for them to function at their peak. So I always like to know how to upregulate gene expression of a gene, but I also like to know what those cofactors are so that I can be sure that they are present simultaneously. So GST is one of the major phase 2 detoxification enzymes. So in this example here, we've got some barbecued steak, which will be containing benzoapyrene and other nasties which have to be detoxified. This diagram here quite simply shows that that xenobiotic needs to conjugate with glutathione in order to be removed from the system. But the glutathione is useless on its own unless we have the presence of glutathione S transferase, because what that enzyme is doing is it's conjugating the glutathione to the xenobiotic and producing a conjugate. So if you're thinking that administering glutathione is all you need to do, you're only solving half of the problem, because we certainly need to get that GST enzyme upregulated there as well. There's a whole host of different GST enzymes. So GST SNPs and oxidative stress polymorphisms combined can actually enhance cancer risk. So now we're starting to look at some of the combinations. So this study examined a combined effect of SNPs in some nitric oxide synthase enzymes or genes and GSTT1, and what they discovered was that genetically driven dysfunction in oxidative stress and pro-inflammatory pathways could augment the risk in susceptible individuals. So the more combinations of genes we can understand, the better the information we have. Now here's one that I think is a bit interesting, because you may not think that a phase 2 detox enzyme polymorphism would have much to do with essential hypertension, but in fact it showed in this case that it increased the risk. So the association between GSTT1 null phenotype and essential hypertension was confirmed in the overall population in women but not men. And in this study, this was with Italian female patients. What we don't know is Is that relationship true just for the Italian population? It could be a sex-specific function as well. We need to take those things into account. So this is where this research is going very rapidly, and the more of this sort of information we have access to, the better our ability to tailor programs to the particular patient group we're dealing with or individual. Back to the recommended dietary intakes. Now, interestingly enough, the Phase 2 detox, GST, is closely tied up with vitamin C intake. We don't fully understand the mechanism of this, but what we've observed is this, that in patients or subjects who have GST deletions and low vitamin C levels, they get increased iron and LDL than those carrying the functional gene variant. And so vitamin C levels are 20% lower in subjects with both GST SNPs. And a deficiency in GST activity might lead to greater reliance on dietary vitamin C. So perhaps those patients should really be having additional vitamin C over the RDI level. And the RDI for vitamin C protects against serum ascorbate deficiency regardless of genotype. Now we're adding another layer of personalisation. So we know a fair bit. There's a lot of things we don't know. So we know a bit about personalised nutrition. We're starting to get a handle on this omics approach, and there is now technology which helps us to get more information from this omics approach. And I think it's changing the future of nutrition science. What do we not know? So we need lots more research. We need more clarification on the relationships between diseases and genes, although, having said that, this is not classical genetics. We're really more interested in understanding what's going on at the cellular level. How does a particular SNP interfere with the inflammatory pathways or the detox pathways and so on? We need to do more studies on gene-food simulation to see what actually happens. And of course, we've got to consider a whole range of environmental factors. This conference looked at my diet, my phenotype, my genes, what is the risk? Health authorities are concerned that if we intervene too early in the NUTRIGENOMICS process, we may increase the patient risk. But what this study was saying, or what this conference was concluding, is that unlike personalised medicine, where genotype data are linked to the risk of developing a disease, in personalised nutrition, the genetic data relate to the optimal diet for a given genotype to reduce disease risk factors. And there are few ethical and legal issues in personalised nutrition. If you want to be an early adopter, there's a hot tip: kill as few patients as possible. And rule number 1, do no harm. So what are we really trying to achieve here? What I think we're trying to achieve is we're trying to tailor diets and nutritional interventions to suit the individual patient. We may do that through an indirect effect, so we may, through a direct effect, so we may actually be providing a nutrient. So directly, if the patient's short on selenium and I provide them with selenium as a supplement or as a dietary element, I know I'm going to upregulate glutathione peroxidase and glutathione S-transferase and so on. I might want to do that indirectly, so I know that if I want to actually increase the glutathione S-transferase gene expression, I can do that by using sulforaphane, the broccoli-derived compound, which activates NRF2 signalling, and we know that increases GST gene expression. If I do that directly and indirectly, I'm giving the selenium cofactor to ensure that I get maximum gene expression. And from a metabolic effect, sometimes metabolites are being broken down. So I might have a polyphenol, I might be having a grapeseed extract, and that's a polyphenolic compound. Those big bulky polyphenols are too large to get into the bloodstream, but they get metabolised in the gut, and those small metabolites may be activating a whole host of signal transduction pathways about which we know very little at this stage. So essentially, diet-derived compounds are having a constant conversation with the DNA in our cells. A lot of this is occurring through the NRF2 pathway, which is a master factor in regulating gene expression in terms of cytoprotection. And in some studies, they show that it's the key to cancer prevention because the mechanism, as we'll see in a moment, is such that there is very strong evidence that we get increased expression of around 2,000 protective genes in terms of disease prevention. This is not cancer therapy, this is cancer prevention. And the evidence is growing that this is a primary mechanism that's not just protecting against cancer, it's protecting against all chronic disease. A paper from 2004 was early on in saying that the NRF2 pathway upregulates many protective detox and antioxidant genes, acting synergistically to increase the efficiency of the cell defence system, and as such has been tagged the master regulator of cytoprotective genes. So this is a really core upstream element in enhancing cell function. How does NRF2 activate cell defences? Well, we have a pro-oxidant signal of some description. It could be exercise. You could run down the road, generate a lot of superoxide because of the exercise, and that's going to be detected by the KEAP1 in the cytoplasm. It could be the sulforaphane from a broccoli meal that could do that. The whole host of weak pro-oxidant signals that activate this pathway. So KEAP1 acts like the monitor and it's picking up the signals, and when it picks up a stress signal, that's the thing that then tells the cell that it needs to upregulate defences, and so then NRF2 translocates off into the nucleus, lines itself up against the antioxidant response element, which is just a series of nucleotide bases on a particular gene. Out of that, we switch on about 2,000 odd cytoprotective genes all in one go, and the sulforaphane from broccoli is the most potent phytochemical activator of NRF2 at this stage. So in terms of prevention against cancer, we know that the phase 2 genes play a very important role, and we know that in healthy cells phase 2 genes are upregulated, and we know that in cancer cells phase 2 genes are downregulated. We also know that the phase 1 genes are downregulated in the healthy cell and upregulated in the cancer cell. So these are genomic expressions that we can change nutrigenomically. I won't go through all this in detail, but there are a number of core cell processes which are part of the NRF2 target genes. But essentially, NRF2 regulates and controls and activates metal sequestrants. So if you want to bind your heavy metals, you need to upregulate ferritins and metallothioneins. You do that through NRF2. There's the redox modulators, the glutathione synthesizing enzymes, the detoxifiers, other cytoprotectives, and interestingly the vitamin D receptor is one of the NRF2 target genes, and if you're having problems with patients' vitamin D levels not being able to be elevated adequately in the blood, I'd suggest you look at upregulating the vitamin D receptor, and then you'll find you don't need those particularly high levels of vitamin D. So my background as a biochemist means that I tend to focus at this level, and I find that to get a really good understanding of what's happening with nutrigenetics, I can't overestimate the importance of understanding what those pathways are that are underpinning all of this. So we looked at that diagram before, which is looking at the way all of these pathways are interactive. Nothing acts alone, and I particularly want to point out a couple of relationships here. So we know energy production, if it's impaired, is going to impair ATP synthesis. With detoxification, if we've got elevated phase 1 pathways as a cancer patient has, we need to try and intervene and deactivate that. Inflammation and oxidative stress have a reciprocal relationship where they continue to feed each other. If methylation pathways are defective and we're producing elevated levels of homocysteine, that is a very potent pro-oxidant compound, and if you're trying to deal with oxidative stress, you've got to get that homocysteine out of the way because it's just feeding oxidation, inflammation, cycling pathways. Endothelial function, well, if that's not operating normally, it's affecting circulation, which affects everything else. And pancreatic beta cell function is affecting everything else too, if that's abnormal. And so on. So part of what's necessary to fully understand what's going on at a cellular level is to understand these interactive processes. So here's the one I want to point out to you. So there's just the Krebs cycle, you've seen it before. The rate-limiting and first enzyme in that pathway is a aconitase, and that's an iron-containing enzyme. The superoxide radical inhibits aconitase. So if you have a patient who has a high level of oxidative stress or redox imbalance, their aconitase enzyme is going to be inhibited, and as a consequence of that, what implications is that going to have for the energy status of that patient? And not only energy status, but a whole host of other pathways which are dependent on ATP synthesis, like a lot of membrane transporters are reliant on a steady supply of ATP. So this superoxide is a core upstream process that we have to get a handle on pretty early in the process. And you see, if you've got a patient who's got an MN-SOD polymorphism, what might that mean for that person? Here's another example of the same thing. So in yellow there, that's the superoxide radical within the mitochondrion. And you've got coenzyme Q10 being generated here, but it's completely incapable of quenching superoxide radical if it's excessive. If that inactivates the aconitase, now you're going to release iron because it's an iron-containing enzyme, and iron and hydrogen peroxide is going to produce the hydroxyl radical, and that bombs out the glutathione. So you can see all of this is closely interrelated, and if you're trying to sort out glutathione status in an at-risk patient, you can't do it just by providing glutathione. You've got to consider so many other factors. So here's an example of a case of estrogen metabolism. So this is what we might do clinically. So we've got a nutrigenetic profile of our patient. We've got a range of SNPs there which we think are at risk. We've got some nutrigenomic interventions that we can use to reduce the inhibition of the CYP1B1, for example, and activate COMT and so on. We've got some test pairing, so in the case of this estrogen case, we might want to do some other urinary metabolites or some other blood testing to have a look at that. And then we've got some specific dietary interventions we might use. Now, if we're trying to detoxify estrogens, the primary aim of the exercise is to stop the formation of DNA adducts, which are highly mutagenic and going to produce cancer of some form. So that's our goal with our estrogen detoxification. So the phase 1 is largely governed by CYP1B1, and then what happens is the product, the estradiol hydroxylated product from that is now taken by the COMT gene / enzyme, and that COMT enzyme now methylates and creates a methoxyestrogen, which no longer has this estrogenic activity. It's glucuronidated by an enzyme called UDP, glucuronyl transferase, and ultimately it's eliminated. Now, whatever COMT doesn't pick up continues to go along the chain, and now glutathione transferase, GST, that we saw before, that's the phase 2 enzyme that now uses glutathione to conjugate what COMT missed, so we're going to produce glutathione conjugates. And what still didn't get picked up there and is just about to hit the DNA is going to be picked up by quinone reductase, which is another phase 2 enzyme. The intermediate stage there is going to produce even more superoxide, and so we use MN-SOD to deal with that. So that, in a nutshell, is how we deal with this process. What can we do to influence this? Well, we know that CYP1B1 is inhibited by diindolyl methane, so you might want to down-regulate CYP1B1 with that. You might want to use methylating agents to deal with the COMT. And then you've got sulforaphane, which actually upregulates all of the remaining phase 2 enzymes. So you can successfully deal with oestrogen metabolism in that way, just showing that sulforaphane is very potent as an activator of quinone reductase. And some of the other things that we've traditionally used in the past for upregulating phase 2, like St Mary's thistle, really have limited effect. So COMT we don't normally think of as a phase 2 gene, but it actually is because of its role in this capacity. And quinone reductase is one of the most amazingly varied hormones, because not only is it essential for oestrogen metabolism, pardon me, and preventing DNA adducts, but it also prevents dopamine from becoming oxidised and adrenaline. It inactivates a whole host of other toxins, and it recycles vitamin E and CoQ10, and it activates vitamin K. I've run over time. Do I have a few more minutes? Do I have a few more minutes?
2 more minutes.
2 more minutes, okay. So let's just skip quickly through some of the gene-diet interactions and looking at the combination. So in this study, they looked at the relationship between CYP1B1 and the GSTs and breast cancer, and they found that the individual SNPs didn't really have any impact on disease risk, but when you put them together, you got a 4-fold increase in risk. So those are all common genes which you can test using a nutrigenetic profile. This other one here is looking at relative imbalances, and it shows that in normal breast tissue, the CYP19, which is the aromatase, tends to be downregulated, and so does the CYP1B1, but the protective enzymes, COMT and quinone reductase, tend to be upregulated. Again, the combinations are more powerful. This one's similar, but they added MN-SOD into this particular study, and they showed that MN-SOD, COMT and the GSTs are part of the protective profile. So if you've got a patient with SNPs which are abnormal in favour of breast cancer, you know that that's someone who's at really high risk. So our translational nutrigenomics, we went through these processes before, looking at the SNPs, looking at the biochemistry, and looking at dietary interventions. I won't go through this one in detail. So in terms of the case management with this particular patient, we've got some intervention tools that we can use which can down-regulate various SNPs and up-regulate others. We can monitor the process with our tests, and we can use the specific interventions. So if we ask the question, are we there yet? I have to ask you to weigh up what you've heard and make your own decision on that. And I'd like to thank you very much for your attention.